Hey guys, this is Miss Bailey. I'm just coming to you with a quick summary of section 3, but also some instructions of how to get through the worksheets that I posted on Teams. This video does not replace you reading section 3. So you still have to read section 3 because it goes more in depth than I do about the material for this section. So our goals for today are that one, we should be able to compare and contrast asexual and sexual reproduction and explain the advantages and disadvantages of each. And the second thing we're going to do is like investigate Punnett squares and figure out how they're used to predict the genetic makeup of organisms. So we're going to have to go all the way back to section 1. Section 1 is where we talked about heredity and traits. And, just remember that heredity is a process of which traits are passed from parents to offspring, and traits are the characteristics that we see in organisms, and that is determined by their genetic makeup. Chromosomes and genes are in section 2, but they also come back in section 3 as well. But just remember that genes are segments of DNA that pretty much govern specific traits. The genes are contained in chromosomes, and chromosomes are found in the nucleus. So, organisms reproduce either sexually or asexually. For asexual reproduction, there's only one parent involved. And then for sexual reproduction, there are two parents whose genetic material pretty much combine to form this new organism. So, like, we're an example of organisms that are reproduced sexually. So, for asexual reproduction, because there's only one parent involved, the genetic makeup of the offspring is identical to that of the parent. And bacteria is an example of an organism that reproduces asexually. In sexual reproduction, we need an egg cell and a sperm cell. Both of these cells containing half of the chromosomes that make up humans. And in total, we have 46, but we get half from one parent and half from the other. Unlike asexual reproduction, in sexual reproduction, the organisms that are the offsprings are not identical to their parent. They're more diverse because, well, you have two parent genes who are like coming together to make these new offsprings. So genes and alleles are in section 3. So, so genes and allele are in section 3. So genes pretty much control traits. Different forms of genes are called alleles, and alleles control the inheritance of traits. Alleles can either be dominant or recessive. So when you have a dominant allele present, that is the trait that will show up. If you have a dominant and a recessive allele, the dominant trait masks the recessive trait, so the dominant trait still shows up. The only time that a recessive trait will show up is if you have two recessive alleles. So probability is the likelihood of something happening, right? In this case, like, what is the chance of an offspring inheriting certain traits from their parents? So Punnett squares is pretty much a chart that shows all the possible combinations of alleles that can result from the genetic cross. The alleles from one parent will be written across the top, right? So if you look at this chart to the left, at the top, these are the alleles from one parent. Along the side are the alleles for another parent. And then the combination is what goes on the inside. And then this determines what percent of the offsprings will inherit a certain trait. And your lab manual walks you through a more in-depth example of how to figure this out. Some useful terms to know when you're working on your assignment is that a phenotype is a physical trait that we see. A genotype is a genetic makeup. So the genotype determines the phenotype. When an organism has two identical alleles, that's called homozygous. So for example, if we're using the letter T to represent tall, right? If there are two uppercase T's or two lowercase t's, those are examples of homozygous alleles. If you have one uppercase T and one lowercase t, that's an example of a heterozygous allele combination. So in terms of today's worksheet, part A, you're going to just be looking at the different organisms that are in each table. And then you're going to highlight the best choice depending on if that organism is reproduced sexually or asexually. So for example, for the first one, we have a grizzly bear and an amoeba, and it tells you whether each is reproduced sexually or asexually. So if you go down the column on the left, it's first asked about the number of parents, right? So for each different type of reproduction, how many parents are required? So for example, if this grizzly bear is reproduced sexually, then you know we need 2 parents, right? We need to use egg and sperm, and then the offspring, they're not genetically identical to the parent. If we come over here to the amoeba, then we know if it's reproduced asexually, there's only 1 parent, there's no need for egg and sperm, and then the offsprings will be genetically identical to the parent. The last row asks about the complexity of organisms. So you can read about this in your lab manual and figure out, if these organisms are simple or are complex. The second part is just using what you learned in the different charts, and there are several charts, I just showed you one, to come up with conclusions. So what are 3 facts that we know about organisms that reproduce sexually? What are 3 facts we know about organisms that reproduce asexually? And then, because it's really hard to type in a Venn diagram, I just gave you a table, but you're just comparing and contrasting them. What are some differences? What are things that are particular to both? And then to each individual ones. In this chart, you would just write the pros and cons. So what are the advantages of asexual reproduction and what are the disadvantages of it? And then you do the same thing for sexual reproduction. What are the pros? What are the cons? In the last part of the activity, we're going to be using Punnett squares to determine the probability of offsprings inheriting certain traits from their parents. For both sections of the activity, I did insert additional video links for more information. So your lab manual should be your primary source. Also watch the videos that I linked in, and if you don't get through the worksheet today, that's completely fine. Just bring whatever questions you have to class tomorrow. So again, by tomorrow, you should have already read section 3. And when we log in for class, we're going to have a quick discussion about the material. And then you will get some time to ask questions and further work on the worksheets. So in the meantime, if you have any other questions, don't hesitate to email me or send me a message on Teams. And I will see you guys tomorrow.